Welcome to the deep dive. We take complex subjects and, well, we try to boil them down to what you really need to know.
Get you genuinely informed.
Exactly. And today, we're tackling something absolutely central to life. Protein metabolism. I mean, just think about it for a second.
Yeah.
Almost every single thing your body does, from, I don't know, thinking to moving to fighting off a cold, it all relies on proteins.
Thousands and thousands of different kinds.
So our goal today is to, let's say, give you a bit of a shortcut through a key part of this Chapter 27 of Lehninger Principles of Biochemistry. We want to unpack that amazing journey.
You mean how our genetic information, that DNA blueprint, actually gets turned into all those different proteins a cell needs?
Precisely. How it happens, moment by moment, inside your cells.
Well, it's a journey that involves just incredible complexity and precision. It's not just making them.
No.
No, it's everything. Synthesizing them correctly, sure, but then modifying them, sending them to exactly the right place in the cell.
Like cellular mail sorting.
Kind of. And then, just as importantly, breaking them down when they're old or damaged or just not needed anymore. It's this whole balancing act.
That's proteostasis, right? That term.
That's the one, proteostasis, keeping everything in a steady state. It's like constant quality control and inventory management all rolled into one.
It sounds incredibly efficient, but also maybe energetically expensive.
Oh, massively expensive.
Yeah.
That's one of the really surprising things we'll get into. It tells a huge story about evolution and, well, just how remarkable these molecular machines are.
Okay, I'm hooked. Let's start at the foundation then. The blueprint itself, the genetic code.
Perfect place to start.
So the genetic code, this is basically the language, isn't it? The dictionary that translates the information stored in our DNA, and then its copy, RNA.
Into the specific sequence of amino acids for each protein.
Yeah.
Yeah, it's the fundamental instruction manual.
And you mentioned it's expensive. How expensive are we talking?
Well, the source, Lehninger, points out that handling this information, specifically making proteins based on it, uses up a colossal amount of cellular energy.
More than other processes.
Way more. For many cells, it can be up to 90% of their entire biosynthetic energy budget.
90% just making proteins. That's unbelievable.
It is. And here's the key thing. A lot of that energy isn't just for snapping the amino acids together, forming those peptide bonds. It's for ensuring the correct sequence.
So it's the cost of accuracy, the information itself.
Exactly. Biology pays a huge premium for getting the information right. Every single amino acid has to be in the right place. Think about it. Each peptide bond costs the equivalent of more than 4 high-energy phosphate bonds NTPs. That's a massive investment per link.
Just to avoid typos, essentially, because one wrong amino acid could ruin the whole protein.
Precisely. So let's break down the code's key features. First, it's a triplet code.
Meaning 3 nucleotide bases specify 1 amino acid, a codon.
Right. Second, it's non-overlapping. The codons are read one after another, like words in a sentence, with no shared letters between them.
Which means if you accidentally insert or delete just one base,
You shift the whole reading frame. Everything downstream becomes nonsense. Total gibberish. That's why the starting point is so critical.
and that's the third point, right? A specific first codon sets that reading frame.
Usually AUG. It tells the ribosome exactly where to start reading the message.
It's amazing how they figured all this out back in the 60s, wasn't it?
Yeah, a real golden age of molecular biology. People like Nirenberg and Mathai did these, well, frankly, brilliant experiments. They made artificial RNA, like just a string of U's poly-U.
And fed it to the protein-making machinery.
Yep. And out came a protein made only of phenylalanine. So boom, you UG codes with phenylalanine. Then Karana used more complex repeating sequences to nail down the rest. It was like cracking an alien language.
So AUG starts things off. Are there specific signals to stop, too?
Absolutely. We have termination codons, or stop codons, UAA, UAG, and UGA. They don't code for an amino acid. They just signal end of protein here.
And that uninterrupted stretch between the start and stop, that's the open reading frame, the ORF.
Exactly. That's usually the part of the gene that actually encodes the protein sequence.
Okay, so it's specific, but you also hear the code is degenerate. That sounds bad, like it's flawed.
Yeah, no, not at all. It sounds counterintuitive, but degeneracy is actually a key design feature. It just means that most amino acids are specified by more than one codon.
Yeah. So multiple words can mean the same thing, the same amino acid, like alanine having 4 different codons.
Precisely. But importantly, each codon only specifies one amino acid. There's no ambiguity going that way.
Okay, multiple codons for one amino acid. How does the cell handle that efficiently? Does it need a separate tRNA, one of those adapter molecules, for every single codon?
Good question. And the answer is mostly no, thanks to something called Crick's wobble hypothesis. It's really elegant. Yeah, wobble. It describes how the pairing between the third base of the mRNA codon and the corresponding base on the tRNA's anticodon loop can be a bit loose, less strict than the first two positions.
So the third position can wobble a bit in its pairing.
Exactly. This means a single tRNA molecule can often recognize and bind to multiple codons that code for the same amino acid as long as the first two bases match correctly. Like a tRNA with a special base called inosinate in its anticodon can pair with U, C, or A in that third codon position.
Clever. So the cell doesn't need 61 different tRNAs, one for every possible code and codon. It can get away with fewer.
Right. It balances speed and accuracy. It's efficient. And this degeneracy, combined with the wobble, has another huge benefit: resistance to mutations.
How so?
Well, think about it. If you have a random mutation, a single base change, particularly in that third wobble position.
Or might just change the codon to another codon for the same amino acid.
Exactly. A silent mutation. It has no effect on the protein sequence. And even if it does change the amino acid, because similar codons often code for chemically similar amino acids, the change might be very minor. A conservative substitution.
So the code itself has built-in redundancy, buffering against errors. It's been streamlined by evolution.
Beautifully streamlined. And while we call it the universal genetic code because it's so conserved across almost all life,
There are always exceptions in biology, aren't there?
always, there are minor variations, especially in mitochondria or some microbes. Like UGA is usually a stop codon, but in our mitochondria it actually codes for tryptophan. Little quirks.
And sometimes the reading process itself gets quirky. I think I read about frame shifting.
Right, translational frame shifting. It's rare, but sometimes the ribosome literally slips, maybe one base forward or backward, and starts reading the rest of the mRNA in a different frame.
Producing a totally different protein sequence from that point on.
Or sometimes a related but distinct protein. Retroviruses actually use this deliberately to make multiple proteins from overlapping genes on a single mRNA. It's like a hidden layer of information coding.
Wow. And then there's RNA editing, too, changing the message after it's been copied from DNA.
Yeah, another layer of complexity. The mRNA molecule itself can be chemically altered before it gets translated. Enzymes can swap bases, or even insert or delete bases in some organisms.
Leading to different protein versions from the same gene.
Exactly. Like in humans, there's a gene involved in lipid transport. In the liver, it makes one protein. But in the intestine, an enzyme edits the mRNA, changing one base to create an early stop codon. Result: a much shorter specialized protein just for the intestine. It adds incredible versatility.
So given all this natural variation in editing, it begs the question, can we artificially expand the code?
People are doing exactly that. Lehninger mentioned the natural and unnatural expansion. Naturally, you have things like selenocysteine and pyrrolysine, sometimes called the 21st and 22nd amino acids.
Incorporated using special tRNAs at what are normally stop codons.
Right, under specific conditions. But scientists have hijacked this idea. They've engineered new tRNA synthetase pairs in the lab to incorporate totally unnatural amino acids with unique chemical properties directly into proteins.
Giving us new tools to study proteins, maybe even design new functions.
Precisely. It's a really exciting frontier in synthetic biology and protein engineering.
Okay, so we've got the blueprint, the code, its nuances, even ways to expand it. Let's move to the factory floor now. How are these proteins actually built?
Right. The synthesis process. It happens in 5 main stages. Think: activation, initiation, elongation, termination, and then folding and processing.
And the main machine doing the building is the ribosome.
Ribosome, yes. It's the huge complex assembly of ribosomal RNA, rRNA, and proteins, a real supermolecular machine. But here's the kicker, the really groundbreaking discovery from around the year 2000.
What's that?
The ribosome is actually a ribozyme.
Meaning the RNA itself is the catalyst, not the protein parts.
Exactly. Specifically, the large rRNA component, like the 23S RNA in bacteria. It's the RNA that physically catalyzes the formation of the peptide bonds between amino acids. There's a huge shift in thinking RNA isn't just passive information carrier. It can be an enzyme, too.
Mind-blowing. And bacterial versus eukaryotic ribosomes are a bit different, size-wise.
Yeah, bacterial ones are smaller, called 70S ribosomes. Ours in eukaryotes are larger, 80S, and a bit more complex.
Okay, so the ribosome is the factory. What about the delivery trucks bringing the right parts, the tRNAs?
Yes, the tRNAs, the crucial adapters. They have this very recognizable shape, like a clover leaf flattened out, but in 3D it's more like a twisted L.
And they have two key ends, right?
Yep. One end, the amino acid arm, is where the specific amino acid gets attached. The other end has the anticodon arm, with 3 bases that pair up with the matching codon on the mRNA. They're the physical link between the nucleic acid code and the amino acid sequence.
So stage 1 must be getting the right amino acid onto the right tRNA. That's activation.
Exactly. Stage 1. Activation of amino acids. This is absolutely critical for accuracy. The enzymes responsible are called aminoacyl tRNA synthetases.
And each one has to be super specific.
Incredibly specific. There's generally one synthetase for each amino acid. And it has to recognize both the correct amino acid and all the correct tRNAs for that amino acid. If it makes a mistake here,
The wrong amino acid gets put into the protein no matter how perfectly the ribosome reads the code later.
precisely. So these synthetases have amazing proofreading abilities. They often have a second editing site. If they accidentally attach the wrong amino acid, say valine, instead of the very similar isoleucine, they can double-check and chop it off before releasing the incorrect tRNA. It's like a two-step verification.
Wow. So much checking and double checking.
It has to be. This interaction between the synthetase and its tRNA is so crucial for fidelity that it's sometimes called the second genetic code. It's not about codon-anticodon pairing, but about the enzyme recognizing the tRNA structure itself.
That makes sense. Okay, amino acids activated and loaded onto tRNAs. Stage 2, initiation, getting things started.
Right. Stage 2, initiation. Proteins are built starting from their amino terminal and N-terminal end. Bacteria use a slightly modified methionine and formylmethionine, FMET, as their very first amino acid, carried by a special initiator, tRNA.
And we use regular methionine.
We use regular methionine, also with a special initiator, tRNA. In bacteria, initiation involves the small ribosomal subunit, 30S, the mRNA, that FMET tRNA, and some protein helpers called initiation factors.
How does the ribosome know exactly where the start codon is on the mRNA?
In bacteria, there's a specific sequence on the mRNA just upstream of the AUG start codon called the Shine-Dalgarno sequence. It base pairs directly with a complementary sequence on the 16S RNA of the small ribosomal subunit.
So it lines everything up perfectly, puts the AUG right in the ribosome's starting position, the P site.
Exactly. It ensures the reading frame starts correctly. Eukaryotic initiation is a bit more complex, uses more factors, and often involves the mRNA forming a loop connecting the 5-foot cap and the 3-foot poly-A tail, which seems to help with efficiency.
Okay, initiated. Now the main event. Stage 3, elongation, adding amino acids one by one.
Yes. Stage 3, elongation. This is a repeating cycle with 3 basic steps, and it burns through energy in the form of GTP.
GTP. Okay. What's step 1?
First, an incoming aminoacyl tRNA carrying the next amino acid specified by the codon in the ribosome's A site, acceptor site, binds. It's escorted in by an elongation factor protein, like EF2 in bacteria, carrying GTP. Hydrolyzing that GTP helps ensure the correct tRNA has bound before proceeding. Another checkpoint.
Accuracy again. Then step 2, making the bond.
Step 2. Peptide bond formation. This is where the ribozyme activity kicks in. The RNA catalyzes the transfer of the growing polypeptide chain from the tRNA sitting in the P site, peptidyl site, onto the amino group of the new amino acid attached to the tRNA in the A site.
Stitching the new amino acid onto the chain.
Precisely. Then step 3, translocation. The ribosome shifts exactly one codon down the mRNA. This moves the tRNA that just donated the chain into the E site, exit site, shifts the tRNA holding the newly extended chain from the A site into the P site, and opens up the A site for the next incoming tRNA.
And this movement also costs energy.
Yes, this translocation step requires another elongation factor, EFG in bacteria, and the hydrolysis of another GTP molecule.
So let's tally that up. We had maybe 2 ATP equivalents for activation.
Right, back in stage 1.
And now 2 GTPs hydrolyzed for every single amino acid added during elongation. 1 for binding, 1 for translocation.
That's right. Over 4 high-energy bonds per amino acid incorporated. It's this huge thermodynamic push that drives the synthesis forward and, crucially, maintains that incredible accuracy required to encode specific biological information in the protein sequence.
An enormous investment. Okay, so elongation continues until
the ribosome hits a stop codon, UAA, UAG, or UGA, in the A site. That signals stage 4, termination.
no tRNA matches the stop codons, right?
Correct. Instead, proteins called release factors bind to the A site. They trigger the hydrolysis of the bond, linking the completed polypeptide chain to the tRNA in the P site.
Setting the protein free.
Exactly. The protein floats off, and then the ribosome complex disassembles the subunits separate, ready to start synthesis on another mRNA.
But the journey isn't over for the protein, is it? It's just a floppy chain at this point.
Not at all. We enter stage 5, folding and post-translational processing. That linear chain has to fold into a precise 3D shape to be functional.
And that doesn't always happen spontaneously.
Often it needs help. Cells have protein chaperones and larger machines called chaperonins, like the GROEL-GROES complex, that bind to unfolded or partially folded proteins, preventing them from clumping together incorrectly, aggregating, and helping guide them towards their correct final shape. This folding process itself can also consume ATP energy.
More energy. And besides folding, there are other modifications.
Oh, a huge variety of post-translational modifications. The initial methionine, or F-met in bacteria, might be chopped off. Specific amino acids might get chemically modified phosphorylation, methylation, carboxylation acting like switches.
What else?
Sugars can be added, a glycosylation, especially for proteins destined for the cell surface or secretion. This happens mainly in the ER and Golgi apparatus. Lipid groups like isoprenyl groups can be attached to anchor proteins to membranes.
Like the RAISE proteins involved in cell signaling.
Exactly like RACE. Some proteins are made as inactive precursors, proproteins like proinsulin, and need to be cut, proteolytic processing, to become active. And disulfide bonds can form between cysteine residues, especially to stabilize proteins that function outside the cell. It's a whole toolkit of modifications to fine-tune function and location.
It's incredible how many steps there are just to get one functional protein. And because it's so complex and essential, I guess it's a major target for things that want to disrupt life, like antibiotics.
Absolutely. Protein synthesis is a prime target. Many antibiotics work by exploiting the subtle differences between bacterial 70S and eukaryotic 80S ribosomes.
So they can poison bacterial protein synthesis without harming ours too much.
That's the idea. For example, tetracyclines block the A site on bacterial ribosomes, preventing new tRNAs from binding. Chloramphenicol inhibits the peptidyltransferase step in bacteria. Puromycin mimics a tRNA and causes premature chain termination in both, which is why it's toxic but useful in the lab.
And some really nasty toxins target our ribosomes?
Yes, things like diphtheria toxin and ricin from castor beans are incredibly potent because they enzymatically modify and inactivate eukaryotic ribosomes, shutting down protein synthesis completely.
A chilling reminder of how vital this process is. Okay, so proteins are made, folded, modified. Now, what about getting them to the right place and cleaning up afterwards?
Right, protein targeting and degradation. The cell is highly compartmentalized, especially eukaryotes. You've got the nucleus, mitochondria, ER, Golgi, lysosomes. Proteins made in the cytosol need to get to their correct destinations.
And they have built-in zip codes for that.
Pretty much. These are usually short amino acid sequences called signal sequences. They act as targeting signals.
Directing proteins to the ER or mitochondria or the nucleus.
Exactly. Let's take the ER targeting pathway. If a protein is destined for the ER or secretion or lysosomes, it usually has an N-terminal signal sequence. As this sequence emerges from the ribosome,
Something recognizes it.
yes, a complex called the signal recognition particle, SRP, binds to the signal sequence and the ribosome and actually pauses translation temporarily.
Puts the brakes on. Why?
To give it time to escort the whole ribosome mRNA polypeptide complex to the ER membrane, where it docks with an SRP receptor.
And then?
Then translation resumes, but now the growing polypeptide is threaded through a channel into the ER lumen or inserted into the ER membrane. The signal sequence is usually cleaved off inside the ER.
And inside the ER, more things can happen, like that glycosylation you mentioned.
Right, N-linked glycosylation starts there. Disulfide bonds can form. Then these proteins often travel through the Golgi apparatus for further modifications and sorting. For example, proteins destined for lysosomes get tagged with a special sugar, mannose 6-phosphate, in the Golgi.
A specific tag for a specific destination. What about other places, like mitochondria or the nucleus?
Proteins from mitochondria and chloroplasts also often use N-terminal signal sequences, but they're typically synthesized completely in the cytosol first, kept unfolded by chaperones, and then imported into the organelle.
And the nucleus, that's different again, right? Because the nuclear envelope breaks down during cell division.
Exactly. Proteins destined for the nucleus have nuclear localization sequences, NLSs. And here's the really clever part. Unlike most signal sequences, NLSs are not cleaved off after import.
Why keep them?
So that after the cell divides and the nuclear envelope reforms, those nuclear proteins, now scattered in the cytoplasm, still have their ticket to get back into the nucleus quickly. This import process involves proteins called importans and relies on the RAN-GT pace cycle for energy and directionality.
Nature thinks of everything. Do bacteria do this sort of targeting too?
They do, though simpler. They also use signal sequences and pathways, like the SEC pathway, involving chaperones and AT passes, to get proteins across their membranes or into the periplasmic space.
And cells can also bring proteins in from the outside.
Yes, through receptor-mediated endocytosis. Cells have receptors on their surface that bind specific molecules, like proteins. Binding triggers the membrane to invaginate, often using proteins like clathrin to form a coated pit, which then pinches off internally as a vesicle, bringing the cargo inside.
A way to import nutrients, but
Mhm.
also.
Also, tragically, a route exploited by pathogens. Diphtheria toxin gets in this way. SARS-CoV-2, the virus causing COVID-19, uses its spike protein to bind to receptors like ACE2, triggering endocytosis to enter our cells. It hijacks our own import machinery.
Okay, so we've made proteins, folded them, modified them, sent them where they need to go. But what about when they get old or damaged or are just not needed anymore?
That's where protein degradation comes in. It's the essential cleanup crew, absolutely vital for that proteostasis balance we talked about. It removes faulty proteins and recycles their amino acids.
And proteins don't all last the same amount of time.
Not at all. Their half-lives vary enormously, from minutes for regulatory proteins that need to disappear quickly to days or even weeks for structural proteins. This turnover is tightly controlled.
How does the cell actually break them down?
In bacteria, there are ATP-dependent proteases, enzyme systems like LON and CLPXP that recognize, unfold, and chop up targeted proteins.
And in our cells, eukaryotes.
The main system is the ubiquitin proteasome system. It's incredibly sophisticated.
Ubiquitin. That's a tag.
Exactly. Ubiquitin is a small, highly conserved protein. It gets covalently attached to proteins that are marked for destruction. This tagging usually involves building a chain of multiple ubiquitin molecules.
How does the cell decide which proteins to tag?
That's the job of a three-enzyme cascade. E1, activating enzyme, E2, conjugating enzyme, and crucially, E3 ligases. The E3 ligases provide the specificity, recognizing the actual target protein and facilitating the transfer of ubiquitin onto it. There are hundreds of different E3s, each recognizing different signals or target proteins.
So the E3s are like the foreman pointing out which buildings need demolishing.
That's a great analogy. Once a protein is tagged with a polyubiquitin chain, it's recognized by the 26S proteasome.
Which is?
The proteasome is a huge barrel-shaped protein complex. Think of it like a molecular shredder. It grabs the ubiquitinated protein, uses ATP energy to unfold it, threads it into its central chamber, and chops it up into small peptides.
Recycling the amino acids, what kind of signals tell the E3 ligases tag this one?
It varies. Sometimes it's damage or misfolding. Sometimes it's a specific modification, like phosphorylation. And fascinatingly, for many proteins, the identity of the very first amino acid, the N-terminal residue, is a major signal. This is known as the N-end rule.
The first letter dictates how long the protein lives.
In many cases, yes. Certain N-terminal amino acids act as signals for rapid ubiquitination and degradation, while others confer stability. It's another layer of control over protein lifespan. It really makes you ask, how does the cell juggle all this? Ensuring proteins are there when needed, gone when not.
It's mind-boggling, and when this degradation system messes up, I assume that has consequences.
Huge consequences. Defects in ubiquitination or proteasome function are linked to a vast range of diseases. In cancer, if proteins that normally restrain cell division aren't degraded properly, you get uncontrolled growth.
Makes sense.
Conversely, in many neurodegenerative diseases like Alzheimer's or Parkinson's, you see the accumulation of misfolded proteins that should have been cleared out by the proteasome, forming toxic aggregates.
So understanding this pathway is critical for medicine.
Absolutely. And it's led to new therapeutic strategies. For instance, proteasome inhibitors are now used to treat certain cancers, like multiple myeloma, by essentially clogging up the cancer cell's garbage disposal system.
Incredible. So wrapping this all up, it's been quite a journey. From reading the genetic code
With its triplets, degeneracy, and wobble,
to the energy-hungry synthesis on ribosomes, those amazing ribozymes.
through folding, modifications, getting zip-coded to the right cellular address.
And finally, the controlled destruction by the ubiquitin proteasome system. It's just a constant dynamic flow.
It truly is. An elegant dance of synthesis, function, and removal, all precisely balanced. It's the essence of cellular life, refined over billions of years. When you stop and think about this intricate molecular choreography happening nonstop, trillions of times over inside you right now, well, it's pretty awe-inspiring.
It really is. A symphony inside every cell. Well, thank you for joining us on this deep dive into the world of protein metabolism. We hope it's giving you a new appreciation for these fundamental processes.
Keep asking questions, keep exploring.
And we'll catch you on the next deep dive.